Hello, BISC1323. This is the beginning of recorded lecture 5-4, still talking about the digestive system. So we went through this entire process sort of focusing on human digestion, but now I want to contrast this with the digestive system of other vertebrates, specifically herbivores. So herbivore vertebrates have evolved digestive systems that enable cellulose breakdown or cellulose digestion. So cellulose, you may remember from BISC1323, part of the plant cell wall, and it's really difficult to deal with. So if you want to get energy out of leaves and grass, you need a specialized digestive system, and there are two different ways in which this has evolved. The first one that we're going to talk about is the digestive system of something called ruminant herbivores. And this is going to include cows and goats and sheep and giraffes and deer, but I'm just putting down cows and goats. So here's a goat, and we can see that it begins just like we do. You got a mouth, you got mastication, you got swallowing, you got peristalsis, there's the esophagus, but here's where things are different. So instead of a stomach, there are actually four different stomachs, or a four-chambered stomach, starting with the largest compartment, something called the rumen. Now, after these four chambers of the stomach, it goes to a small intestine, a large intestine, poop. So that part is all sort of normal. What we want to focus on are these four chambers of the stomach, specifically this first one called the rumen. So that's why they're called ruminant herbivores. They have a four-chambered stomach. The first chamber is called the rumen. This is a large chamber where bacteria break down cellulose. So we talked about, again, going back to human digestion, that our large intestine had a lot of different bacteria in it that, broke down fiber and released vitamins and stuff. Well, when it comes to cellulose breakdown, it's also bacteria that are responsible for doing this. But the bacteria are located not in the large intestine, but in this massive rumen chamber. The rumen is so big because, these ruminant herbivores are taking in a lot of plant matter, so you're going to need to house a lot of bacteria to break down all of that cellulose. Another detail to this is that these ruminant herbivores do something called chewing cud. They will regurgitate the contents of this rumen, chew it a second time, and then swallow it again. So this is not throwing up, this is not vomiting, because the rumen doesn't have stomach acid. So the regurgitated contents of the rumen are really just the same leaves and stuff that they chewed, but now it's been mixed with bacteria. This is to make sure that it gets mechanically broken down and mixed up with those bacteria even more. So we define what the rumen is. These ruminant herbivores regurgitate the contents of the rumen to chew again for more complete breakdown, and then after it's swallowed, it goes to the second chamber, the third chamber, the fourth chamber. We're not naming any of those, and then small intestine, large intestine, and then we're done. So the rumen is really where the action is happening. There's another way to do this, though. So our second way of having cellulose breakdown is found in what is creatively called non-ruminant herbivores. And this includes rodents and rabbits and horses and koalas and the panda, but I'm just putting down rodents and rabbits, and we're using bunny rabbit as our example of this. So let's take a look at this digestive system. We've got a mouth, we've got an esophagus, we've got a stomach, small intestine looking pretty normal. Here's the large intestine, also known as the colon, and there's this massive pouch that's part of the large intestine called the cecum. This is where the action is happening. So instead of doing the cellulose breakdown, instead of housing all these bacteria in the beginning, like these ruminant herbivores did, these non-ruminant herbivores are breaking down cellulose close to the end instead of close to the beginning. So these non-ruminant herbivores have a large cecum that is part of the large intestine that houses bacteria that break down cellulose. Now, there's a problem with this. So their digestive tract, other than this pouch here is pretty much the same as what we described last time. And if you remember the functions of all these different parts, the best absorption, the part of this tract that was best at absorbing nutrients and stuff, was the small intestine. And so it kind of made sense to break down cellulose in the beginning, because by the time it made its way to the small intestine, it was ready to absorb. But now we've broken down cellulose at the end. We haven't had a chance to absorb that. So what are we going to do? Well, let's articulate this problem. The best absorption of nutrients is in the small intestine, and this cecum, where the cellulose breakdown occurs, is located after the small intestine. So, okay, maybe you could start puzzling out some solutions, because otherwise you'd be pooping out stuff with a bunch of nutrients in it. Maybe we could reverse the process. No, the muscles don't really work that way. We can't really reverse peristalsis. So our problem is we're generating droppings that have a lot of sugars that have been liberated. They've been, broken down from cellulose, but we haven't had a chance to absorb them yet. Maybe you can guess where we're going with this solution here. So if your droppings contain a lot of nutrients, the solution is to eat those droppings. So not all non-ruminant herbivores do this, but many non-ruminant herbivores, including rabbits, as we're seeing here, do something called coprophagy. So once the food has gone through the digestive tract a single time, it's going to come out as this type of dropping called cecotropes. And again, it's high in nutrients because the cellulose has been broken down, but those sugars that come out of it haven't been absorbed yet. So they're going to eat these, sometimes directly, sometimes they come out and then they eat them, but they're going to give them a second pass through the digestive system so that these nutrients can be absorbed. When they come out the second time as normal feces, these things are not eaten. So coprophagy is what this is called. These animals eat their own feces. And if we want to describe coprophagy a little bit more, they produce two types of droppings. The first type is eaten, again, to give that food a second pass through the small intestine. This is required for proper nutrition. The second type is not eaten. So the last thing in this chapter is to talk about vitamins. I think you know me by now. We're not going to memorize this massive table of vitamins because this isn't even the only table of vitamins. There's also this table of vitamins, and sort of related to vitamins, there are essential minerals. We're not going to memorize this table of minerals. And there's another big table in this section, this table of essential amino acids. We're not going to memorize this table of essential amino acids. But what is the take home from all of this? Well, there's a lot of stuff that we can build ourselves. We can build proteins, we can build fats, but we cannot build these vitamins from scratch. They've all got, complex molecular structures. We cannot construct these through our own metabolic pathways. So we need to acquire these vitamins as part of our diet. Similarly, we cannot build iron from scratch. I mean, it's an atom, so obviously we can't build it from scratch. Same thing with all of these other things. So these minerals are also something that is required as part of our diet. And when it comes to these amino acids, remember there are 20 amino acids out there. These are the building blocks of proteins. There are actually some of them that we can build from scratch, but there are others that we cannot build from scratch, so we have to get these amino acids as part of our diet. So the take-home message is things that we call vitamins, certain minerals, and what we call essential amino acids must be acquired from our food. They cannot be made by the human body. Okay. That closes out the digestive system chapter. We're not done with this recorded lecture though, because we're going to move on to the next one, going from poop to pee, as we talk about osmotic regulation. This is going to involve urine and all of that stuff. Now, the reason this chapter is not called the urinary system and instead is called osmotic regulation and excretion is because the urinary system is part of a larger big picture process about osmotic regulation. So, what does that even mean? Well, let's review some terminology, why don't we? Way back from BISC1323, let's look at the term osmosis. I'm going to read the key terms definition, but this should be a familiar looking figure. Osmosis is the movement of water through a semi-permeable membrane from an area of low solute concentration to an area of higher solute concentration. So water is going to move from where there's not a lot of dissolved things to an area where there is a bunch of dissolved things. This can be into the cell or this can be out of the cell. It all depends on where the dissolved stuff is. You may remember from BISC 1303 that there are several different possibilities for what can happen to a cell. If the cell is in balance with its surroundings, you're going to have no net movement of water. These are some healthy looking red blood cells. But if there are too many solutes in the surroundings, the water is going to exit the cell. They're going to shrivel up. That's bad for them. And if there aren't enough solutes in the surrounding fluid, water is going to enter the cell because there's more stuff in the cell, and they're going to swell up and start bursting. So the purpose of this entire chapter here is to ensure this kind of balance. It is important for our blood specifically, but also our cerebrospinal fluid and interstitial fluid, all the fluids in our body, to be an isotonic solution where things are in balance. This means the surroundings have the same amount of solutes as the inside of the cell. We're going to achieve this balance by getting rid of extra water and getting rid of extra ions, whatever it takes to achieve this balance. So, the way we get rid of this stuff is going to involve the kidneys. There are two kidneys located in the lower back, and so we're obviously focused on human urinary systems here. We're gonna compare and contrast this at the end of the chapter. Whether it's male or female, two kidneys sort of in the lower back. If we zoom in on a kidney, we're gonna see. Here's a zoom in. If we zoom in even further. Here, look at this little structure here. That's called a nephron. And if we zoom in even further, here's what the nephron looks like. So before I go through the detail of this, I want to note that each kidney has about 1 million of these structures. They're going to do the actual job, but they're part of this larger structure, the kidney. So 2 kidneys in the lower back, each one has about 1 million nephrons. So what is happening? We'll number these steps, as I've done with a lot of things. This is what's happening at a nephron. So blood arrives at the kidney from what's called the renal artery. We see it labeled here. Now, you may remember from a couple of chapters ago that blood comes back to the heart through a vein, but it goes out of the heart through an artery. Veins are at low pressure because that blood is on its way back home. It's coming back to the heart after a long journey. Arteries are at high pressure. They just came from the heart. They're being pumped out with a lot of force. So this blood that is entering the kidney and it's going to be entering these nephrons is at high pressure. So blood arrives at the kidney from the renal artery at high pressure. Now let's zoom in on one of these individual nephrons here. What happens first is this structure called the glomerulus. The walls of the blood vessels in this glomerulus are particularly thin. So blood is coming in here, it's at high pressure, and the walls are thin. What that's going to do, and here we see blood in, this sort of glomerulus structure, blood out, but because it's being pumped in with high pressure, and because the walls of the glomerulus are thin, anything in the blood that is small is going to be filtered out of the blood. Anything in the bloodstream that is big is not going to be filtered out, and it's going to exit right here. It's going to exit the glomerulus. So what do I mean by big and small? Well, let me review this first. Small molecules are going to include, arrives at the renal artery at high pressure. Small molecules like water, glucose, amino acids, urea, we'll talk more about this later, and just various charged ions are going to be forced through the porous walls of the glomerulus. What do I mean by big stuff? Cells and proteins. Cells are too big, proteins are too big, they're not going to be filtered out. They should not end up in the glomerular filtrate. They should, just return to the bloodstream. So if you ever pee in a cup as part of a physical or a doctor's checkup, an annual visit, whatever it is, if you're ever peeing in a cup, they're checking for a lot of things. One of the things that they're checking for is cells and proteins. If you've got cells, blood cells or protein in your urine, something has gone horribly wrong. Some damage to the glomerulus maybe. Sometimes chronic high blood pressure. If the blood is too high, it can start popping these things and you could end up with everything sort of flooding out. But under normal circumstances, you shouldn't have cells and proteins filtered out, but all of the small stuff makes its way out of the bloodstream into what's called this glomerular filtrate. So at this point, the glomerular filtrate, all the small stuff in the water, enters a system of nephron tubes that are surrounded by blood-containing capillaries. So there's our glomerulus, and here's the system of tubes. So here's the filtrate going up and around and then down and then back up again, and we can see the capillaries that are surrounding it. The purpose of this journey is, let's go back to this statement, all the small stuff is filtered out. Some of this small stuff we want to get rid of. Urea, we'll talk about it later, is a waste product. We want to pee this out. Water and ions, again, depending on the balance of things, these are oftentimes wastes. We want to get rid of these. But glucose, that's not a waste product. We don't want to pee this out. Amino acids, those are useful molecules. Those are the building blocks of proteins. We don't want to pee these things out. However, they got filtered out. They got filtered out because they're small. So before this becomes urine, we want to take back all of those useful, good molecules that we want as they pass through this system of nephron tubes. So that's why this is so long, to give a chance for all these useful things to be reabsorbed into the bloodstream. So useful molecules like glucose and amino acids are reabsorbed back into the bloodstream through this renal vein. Sorry about that. The renal vein is going to have to involve us zooming back out again. Oh yeah, there's the renal vein, which all these nephrons are going to connect to and send things back to the bloodstream. And again, we're reabsorbing these useful things. There's a particular structure in the nephron called the loop of Henle. You'll notice it right here. Loop of Henle. It goes down and then it comes back up again. The loop of Henle helps to absorb water. Now, we do want to get rid of some water and ions, but again, it's all about balance. We don't want to pee out all of our water. We don't want to pee out all of our ions. And so this loop of Henle is helping to reabsorb those things. This helps concentrate wastes in urine. And finally, once we've gone through the process of filtering out and reabsorbing, we're left with what we call urine, which is emptying into a collecting duct. There's our collecting duct. Here's the end of the nephron tubes. And again, we've got like 1 million of these in each kidney. All those collecting ducts are going to empty together, going down through a ureter, which is going to lead to a urinary bladder, which the contents of which can be excreted, urine labeled there, through a urethra. So here's the terminology. The nephrons empty their contents, called urine, into a collecting duct that leads through the ureter to the urinary bladder for storage, and then urine is excreted via the urethra. And yep, there's urethra in male and female urinary systems. Yep, that's where we pee this stuff out. Now, this process of filtering the blood, of filtering out small stuff, reabsorbing good small stuff. This is happening constantly because, again, we've got a million of these in each kidney. Your blood is always circulating, your blood is always going through. The entire volume of your blood is filtered out many times per day. So again, this is constantly happening. So that's the main process of in this chapter. But the last thing I want to talk about, again, this is kind of a short chapter, is to talk more about these wastes. What is it that we're peeing out? What is actually exiting the body? So one of the wastes that we want to get rid of is water. Now, hold on a second. I just told you there was this loop of Henle structure whose whole job was to reabsorb water. Well, it's true that we don't want to pee out the entire blood volume's worth of water because we're filtering this constantly. But again, it's all about balance. If we've got too much water, we do need to get rid of some of it. Our blood can only have so much, it only has so much room. There's only so much total blood volume. If we have too much water, we do need to get rid of it. So this is a waste. Similarly, hydrogen ions are a waste. You may remember hydrogen ions are what the pH scale measures. Again, we don't want to get rid of all of these hydrogen ions, then our blood would become too basic. But we don't want to keep all of these hydrogen ions, then our blood would become too acidic. So again, to maintain the pH of blood, these things are eliminated in our urine. Similarly, other charged ions. Again, the loop of Henle is reabsorbing these. We want to maintain balance and homeostasis of these ions, so there are lots of ions that we end up peeing out if we have too many of them. But the most interesting waste that comes out is excess nitrogen. So to understand this, we have to look a little more closely at the whole excess nitrogen waste. So what is nitrogen? Why does it matter? Where does it come from? What is it used for? Well, nitrogen is used in a lot of things, but most notably, nitrogen comes from nucleotides, so DNA and RNA. There's nitrogen in the nitrogenous base. And nitrogen is also found and used in proteins. These are just 5 of the 20 amino acids, but trust me when I tell you, all 20 of those amino acids have at least one nitrogen as part of their structure. So if you're building proteins, which you're always building proteins, and if you're building DNA and RNA, you're always building DNA and RNA, you're going to need nitrogen. And we get this from our diet anytime we eat protein, and you're pretty much always eating protein in most meals, and so that's going to supply you with nitrogen. The issue is we don't really have a way to store nitrogen. So if you eat, a high protein meal, you're going to get in a bunch of nitrogen from that protein. Without a way to store it, you have to get rid of it. So background, amino acids, part of proteins, and nucleotides, part of DNA and RNA, contain nitrogen. If we're breaking these things down, that produces excess nitrogen. So the simplest way to deal with excess nitrogen is a chemical compound called ammonia, or NH3. It's the simplest form of nitrogen. It's really easy to make from any extra nitrogen you may have, but it's toxic. You can't store this in a bladder. It's going to, be toxic to those cells. However, if you're the type of animal that is constantly surrounded by water and you don't need a urinary bladder to store wastes, you could just get rid of this constantly. So fish, for example, do secrete their nitrogenous waste in the form of ammonia. It's not going to be toxic to their body because it's not stored in their body, and it just diffuses through the surrounding water. This is the simplest way to do it. Ammonia is easy to make. You can't store it, but if you can just get rid of it, you can just get rid of it. Fish excrete ammonia into the surrounding water. Okay, obviously there are going to be some other strategies that I'll reveal here. Well, the next one I want to look at is reptiles and birds. Reptiles and birds take their excess nitrogen and use it to build something called uric acid. Now, this is a complicated molecule, and yeah, there is some nitrogen here, but there's also oxygen, there is some carbon. It takes a fair amount of energy to create this uric acid molecule, and we know what's going to happen to it. It's a waste. Just to get rid of this, you're spending energy building something that you're just going to eliminate as waste. There has to be some advantage to this. Well, there is. Uric acid crystallizes, it forms a solid, and it's not toxic. So even though reptiles and birds have to spend energy to build a waste product, they can store it up in a form that's not toxic, they can eliminate it when they want to, and they can eliminate it without having to, have a big, heavy, water-filled bladder. You don't have to dissolve this in water. You could eliminate this in the form of a solid, which is what gives us bird guano. They don't have urine in the same way that we have urine. It's the waste of their digestive system and the waste of their urinary system sort of combined into a mostly solid form, because again, this crystallizes into a solid. This also explains why you can use this as fertilizer. There's a ton of nitrogen in guano, and you know who loves nitrogen? Plants love nitrogen. So yeah, it does make very effective fertilizer. So let me summarize this. Reptiles and birds excrete something called uric acid. It costs a lot of energy to build, but uric acid is solid. It can be eliminated without water loss in the form of what we call guano. Now, there's obviously a middle path here, and that's going to be mammals. Mammals create something called urea. Now, urea is more complicated than ammonia, so it does take some energy to build this molecule. However, it doesn't take as much energy to build as uric acid. This is a much simpler molecule. So again, kind of a middle ground. The trade-off is urea does not crystallize into a solid like uric acid does, so we do have to store this in water. Again, it's not toxic to us, but it does need to be stored in water, and it does need to be eliminated with water in our urine as opposed to in guano. So mammals excrete urea. It costs energy to build, but less energy than making uric acid. It's not solid, so it needs to be eliminated with water in urine. So again, some pros and cons here. It really is sort of a middle path between these other two strategies. Now, the last thing I want to point out in this chapter regarding nitrogen waste, we've talked about uric acid and urea, and something I want to bring up the medical condition gout. I hope that none of you have suffered from this before. I know I haven't, but if you've ever heard of the condition gout, it is the crystallization of uric acid, specifically in these toe joints, in the big toe. It can happen in other parts of the body, but it typically happens here, and it's painful just to look at. You can imagine these crystals in between the bones. I've heard the pain described as exquisite, and so obviously this is a horrible thing to happen, but it's happening because of these uric acid crystals, which should be confusing to you, right? This is obviously a human foot here. This is obviously labeled uric acid crystals, but I just finished telling you that birds and reptiles make uric acid. Mammals are supposed to make urea. Well, I didn't lie to you, but there is a bit of a twist here. For reasons unknown, it's probably an evolutionary mistake. I don't think there's an advantage to this. Mammals do make urea, and humans are mammals. We do make urea, but in addition, humans and some related primates actually do both urea and uric acid. So that explains why these crystals can happen. So mammals as a whole all build urea, but humans and related primates additionally also build some uric acid. And again, the evolutionary advantage to this is not known. It's thought to be a mutation in some metabolic pathway that makes this happen. But yeah, interesting side note that humans in specific do both of these things. Okay, that brings us to the end of this chapter on osmotic regulation.